Inside the neuron, there's a potassium ion that cannot leave the neural membrane, and outside there's a sodium ion that cannot enter the membrane for the resting potential of negative 70 millivolts in a neuron to be maintained. These two ions must remain in their original spots, meaning that the sodium ions cannot diffuse into the membrane because its channel is closed, causing outside the neural membrane to have a net positive charge, and the potassium ions cannot diffuse out of the membrane because its channel is also closed, resulting in a net negative charge inside the membrane. There are other factors such as contributors that help to maintain a resting potential so that the inside of a neuron is still negatively charged. These are large protein molecules and small chloride particles, both found on the inside of the neural membrane. But the most important contributor is the sodium and potassium exchange pump. This helps to maintain the balance both inside and outside the neural membrane by pumping potassium ions that are outside the membrane back into the membrane in twos, and sodium ions that are inside the membrane outside in threes by active transport.
Just tell me what you want.
A stimulus is anything that triggers or causes a reaction. In this case, the stimulus is a toothpick. This stimulus triggers the opening of some sodium ion non-voltage gates, allowing some of the sodium ions to enter inside the membrane. When this happens, this causes the voltage inside the membrane to increase to a certain threshold level. This means that the voltage inside the membrane increases from negative 70 millivolts to negative 54 millivolts. When a threshold level is reached inside the neuron, this allows all the sodium voltage gates to open. All the sodium ions are now able to diffuse inside the cell membrane, causing the negative charge to become positive. This process is known as depolarization. This movement inside the neuron increases the voltage from negative 54 millivolts to positive 35 millivolts, also known as the action potential. Once the movement of sodium ions into the membrane has reached its action potential, all the sodium ion gates close. This then allows for all the potassium ion voltage gates to open and allow potassium ions to diffuse out of the membrane, reducing positivity inside the membrane, causing it to become negatively charged. This movement reverses the result from depolarization through repolarization, so the voltage inside the membrane begins to drop. After repolarization has occurred, the potassium gates begin to close, but slowly. This allows for extra potassium ions to pass through the channels and diffuse outside the membrane, creating an imbalance inside and outside the neural membrane through an overshoot of potassium ions. This causes the membrane voltage to drop even further down to negative 90 millivolts, past its original resting potential voltage. Time it takes to return the neuron to its resting potential of negative 70 millivolts from negative 90 millivolts is called the refractory period, and during this period a nerve impulse does not occur or is not sent. When trying to re-establish the resting potential of the membrane, the sodium-potassium exchange pump is used to return the ions to their original location. For this to occur, the pump uses ATP to actively pump sodium ions out and potassium ions in. So 3 sodium ions are pumped out while 2 potassium ions are pumped into the membrane, thereby re-establishing the original resting potential of negative 70 millivolts, creating a split between the charges inside and outside the neural membrane. The signal sent to the neuron is able to move from one point to another because of the action potential that is formed. Once the depolarization of the membrane occurs, it causes the voltage to increase to an action potential. This motion of impulses acts as a switch that causes the next side of the membrane to become activated until the impulse has been completely sent. The action potential is able to travel in wave-like motions from one adjacent side to another. In a myelinated neuron, when an impulse is sent into the axon, the signals are able to move from a point of the nodes of fiber to another through a jumping motion. This is known as saltatory conduction. In an unmyelinated neuron, the signals sent by the impulse travels in a straight line without any breaks or spaces in between each movement. This kind of impulse transmission is known as a continuous conduction. Individuals with a myelinated neuron are able to receive nerve impulses much faster and efficiently than people with unmyelinated neurons, whose impulse will take longer to reach the brain and the spinal cord. A synapse is the space between neurons that do not touch in the nervous system. As the nerve impulse moves through the axon to the edge of the neuron, the small vesicles found at the edge of the neuron are called neurotransmitters. These releases acetylcholine, an excitatory chemical that diffuses into the second neuron's dendrites. During this process, the signals move at a slower pace than other impulse transmission areas. Once the acetylcholine attaches to the second neuron's dendrites, it opens the sodium channels, allowing sodium ions to diffuse into the second neuron through depolarization. This creates an action potential. This process will only stop once an inhibitor, such as cholinesterase, which is an enzyme, attaches to acetylcholine and begins to break it down. Once it begins to break down acetylcholine, it closes the sodium gates and does not allow sodium ions to diffuse into the second neuron anymore. This is known as repolarization of the neuron. The remnants of the neurotransmitters chemical, acetylcholine and cholinesterase, are then reabsorbed back into the neurons to be stored for when the next impulse is going to be sent.